0.4543, that is parameter of silicon, and this constant. L is the crystallite size and delta omega is this Raman sheet. When you put this value, then we calculate the crystallite size of that silicon left between the pores. And what is the size that coming out? In A, the size is 2.8 nanometer. B, 4.3 nanometer silicon size. And in C, 6.6 nanometer. And now you see, dimension of 3 exciton of whole silicon is 4.9 nanometer. And you see in this sample, see, we don't observe any fluorescence because it is bigger than the Bohr exciton radius of silicon. And then in sample B, we get a little fluorescence. In sample C, we get a very nice fluorescence. And now the question, why this fluorescence is coming? Because in literature, a lot of things that is given. Some people say it is due to some functional group present in the porous silicon. Few says no, it is due to quantum confinement. Then there is a pair to verify that. And how I verify? Let us. So these are a civil model to understand the PL in porous silicon. Here quantum confinement model, surface hydride model, defect model, surface state model. All these might be responsible for fluorescence in porous silicon. What I did? Okay, there is a FIR. All the functional groups are there in all three samples. Okay, and now what I do? I heated the sample. You see, at 100, 200, 300, 400, and at 400, even 500, most of the functional group are dissolved. They are not there. Still, we are getting fluorescence. Hence, all these other possibilities are ruled out by this. So, what left is only quantum confinement effect, which is well established phenomena now in the nanotechnology. And also, as we see, the size of silicon crystal is smaller than the Bohr exciton radius. So you completely clearly see the quantum confinement effect in the porous silicon. Okay. So this porous silicon, to make the porous silicon, you can remove this porous part from the silicon wafer. Immediately you increase the current, and this porous layer will float in the solution. You take that, grind it, and make the nanoparticle. So this is what also I did. Here you can see the CAM analysis of our porous silicon. You can see the pores around 12 nanometer, and the size of the silicon crystal will be in the range of around 2 to 3 nanometer. So we see this here. There is a blue shift in the photoluminescence. It will decrease in the particle side, and here is the silicon nanoparticle side. And this is visual. Okay, now this is one part where we have seen the quantum confinement in a silicon quantum wire, which has smaller size than the Bohr exciton radius. But now if we want to use as a substrate, then we need to quench this photoluminescence that is coming from porous silicon. Otherwise, if we put some material into the pores to study the quantum confinement effect, then there will be an overlapping of photoluminescence of that material you confined into the pores and also porous silicon itself. So the first thing that you need to do, the quenching of this photoluminescence. And how I did? This is again few chemical process that I applied and I quenched the photoluminescence. I took the sample which shows a very nice fluorescence. The pore diameter is 12 nanometer. Crystalline size around 3 nanometer. And what I did? Porous silicon kept into S2O2. What you do by keeping that, you just partially oxidize that porous silicon. The surface of that is partially oxidized. Then, okay.